Hello everyone, this is Chao Yang. I'm a PhD student from UIUC, and today I'm going to present our ICLR 2023 paper, ManyDG, Many Domain Generalization for Healthcare Applications. So this paper is collaborated with Professor M. Brandon Westover from Beth Israel and Deaconess Medical Center and Harvard Medical School, and Professor Jimin Song from UIUC. So here is the outline of my presentation. I will first give a brief background introduction of domain generalization settings, and then apply these settings on the patient level dataset in healthcare domain. Next, I will we will go to the method section, and then I will first introduce the methodologies, the motivations on the data generation process and how we got to design this prediction model, and then comes to the actual many domain generalization method. After the method section, I'm going to introduce some brief experimental results and then conclude these papers briefly. So here is the domain generalization settings illustration. We got a training set and the test set. We wanted to train in models, usually supervised models on the training set, and then test it in the test set. So in this setting, the training set are usually consists of multiple data domains. You can view it as different styles. And for example, in image classification, domain 1 might be some sketch of animals such as dogs and cats. And then domain 2 can be all your pennings of dogs and the cats. And domain 3 can be some other image styles like drawing or some other styles of the dog and cats. And then we wanted to train the models on these several training domains and then test models on the test domains, which is a new style that we haven't seen, like cartoons, dogs and cats. And then we will see that the data samples in domain 1 will usually contains the domain 1 information, the style information, and the label information. Domain 2 will contain the domain 2 style information and label information, etc. for domain 3. But the models we want to optimize, we hope it can only extract the label information, take those label information into use and ignore the style information, and then give the prediction, because we will generalize it to a new domain, to a new style. So the style information is not useful in the prediction part. So that's why we wanted to apply these settings to patient level data set, because it's actually quite similar in this healthcare domain. Many of the patients, they can have different characteristics, such as each patient, they have different genders, ages, and the data collection environment. And we view these different characteristics, they can be viewed as the style, which is treated as covariants that are not relevant to the predicted tasks, such as sleep stage classifications. So in the paper, such as we wanted to do sleep stage classifications for each patient. And then some patients, long EEG brain recordings are collected from the lab, and then some recordings are obtained at home. So their collecting environment are very different, but those environment discrepancies will be reflected on the data. We don't want the models to take those information into use, because they are not theoretically connected to the final labels. We want to remove them. We wanted to remove the disparity of the patient covariance. So that's the goal. We will train a model on a set of patients, and each patient they will generate different data samples. We train the model on those samples, and then we generalize the models to some new patients to obtain labels for these new patients' data samples. Okay. And then this is the motivation of our models. We will introduce a data generative model, which is some kind of assumption on data generation process, and the motivation on build a factorized prediction models to get the true labels. So remember, each patient in our paper or my presentation, each patient is treated as a domain because they are very different. They have different style. And then we assume each data sample X is generated from a two-stage process. The stage one, we sample a domain from a meta-domain distribution. Stage two, we sample a data sample from a distribution conditioned on the domain and the latent class Y. Okay, that's why we use this day sample from this domain meta distribution P, and then samples from this conditional distribution given domain and Y in the latent label. Okay. So assume all these patient data set are generated from this two-stage process. Then if we obtain a new data sample X, what are we gonna do is we're gonna find its latent domain first, and then use the latent domain to help us to predict the final labels. So we build these factorized prediction models by this mathematical formula. This posterior Y P y given X is mathematically equals to an integral of P y Z given X integral over Z. And then this probability term can be factorized into Py given X Z times Pz given X. Okay, I can explain it in detail here. So the factorized prediction models actually inspired us to design this encoder model. You can view the right part, the encoder part, Pz given X means given X, we want to build an encoder model to find its true domain Z. And then after that, we build another predictors to parameterize this Py given X and Z. Means we have this data sample X. We know it's latent domain Z. We want to do the prediction to uncover its true label Y. Okay. So in the lower part, the pipeline illustration, we use two stages to build this encoder model. First stage is very simple. We use this edge neural network to extract a feature representation. We basically map this raw data X to its latent feature representation V. So this V feature representation, it contains both the domain information and the label information. Okay. And then we use a neural network Q to extract its domain. After extraction, we hope this domain factor Z can only contain this domain information. We hope we will have auxiliary models to enforce these things to happen. Okay. And then right now, assume this V contains the domain information plus label information, and the Z contains the domain information. We build this predictor two step. First step, we do orthogonal projection, remove this Z component from V, which mathematically ensure that this V orthogonal Z will only contain label information. And after we get this V orthogonal Z, we build a G neural network to do the label prediction, because this components only contains this label information. Okay. To ensure that the after encoder Q, this domain Z really only contains domain information, we build this reconstruction decoder design. So this design is motivated by data generation process. The formula X sampled from P given day and Y. Okay. So this day is basically the domain, Y is basically the label Y, and then that data generation model tells us, okay, give me Z, give me Y. I can parameterize this probability distribution to a neural network to generate a sample from the patient data and with label Y. Okay, we make this happen, which is that we input two samples to the neural network, X and X prime. These two samples are from the same domain, which is from the same patient. So they have the same domain information, right? These two samples contain the same domain information, and they can potentially contain different label informations. Okay, so let's see the first pipeline, the upper pipeline X, the same one we have shown before. X contains the domain information plus label information initially. We extract its features using H. The feature representation V still contains domain information and label information. And we use this Q neural network to get the domain representation Z. And then the cool things happening. We use Z, the domain information, plus actually in implementation, we use concatenation. This Z concatenate the label of the sample X prime to reconstruct the sample X prime. Actually, here we reconstruct the feature representation of this X prime, okay, by another neural network P. So this is actually valid because you can view, okay, the feature representation of V prime, what information it contains. It contains the domain information. Okay, that information is provided by Z. It contains the label information. Okay, that information is provided by Y prime because Y prime is its true label, right? And we call that the decoder part. This will ensure, after this reconstruction, it will ensure that the domain information Z, it actually only contains the domain information. It will not contain label information because label is already provided by Y prime. And it will be better if X and X prime initially have is of different labels. Then even if they contains the label information of X, it's not useful in the reconstruction part because X and X prime, they have different labels. But in the experiment, we doesn't differentiate the labels of X and X prime empirically. Both ways works fine. So we combine the motivation from the factorized prediction model and the motivation from the data generation models together to build these two pipeline model. Okay, so in the training, we input two samples from the same domain or the same patient at one time, and then we follow this two-stage pipeline, two-step pipeline. Step one, the feature extracting. Step two, domain encoder. Step three, pre-step orthogonal projections, and then next step is a label prediction. And in the meantime, we do mutual reconstruction from X to X prime, from X prime to X to enforce this domain factor Z only contains the domain information. Okay, use a Siamese type architecture. But in the inference, we just use one part of the pipeline and then go through these three steps to do label prediction. Okay. And here is the part of our experimental results. We do experiments on two healthcare monitoring data sets, which is the biosignal data set, the EEG data set for seizure detection, EEG data set for sleep staging. And we also apply it onto electronic health record data sets. We apply it on EHR sequences for drug recommendation and readmission prediction. And we compare with many different domain generalization models. Yeah, I will not introduce these here, but our performance is generally better than these baseline models. So here is a conclusion of the paper. So in this paper, we introduced the many domain generalization, ManyDG problem in healthcare. We call it many because in the healthcare data sets, we treat each patient as a separate domain. And then usually for domain generalization problems, people use like 4 or 6 to 5, 6 domains. But in our data sets, we can have hundreds of thousands of patients. So we are dealing with much more domains than previous settings. And yeah, here's the illustration I've shown before. And in the experiment, we have shown better results using our ManyDG methods compared with existing domain generalization models. Our model is more scalable to much more domains. and we perform better on healthcare monitoring data sets as well as electronic health record data sets. Thank you very much for listening, and I have attached the GitHub repo of the paper and my email for your reference in case you have any problems to discuss. Okay, thank you very much.